Hello everyone, welcome back to Swati's Bioscience. So in last few sessions we have been discussing about the cellular respiration in aerobic conditions. Okay, and the cellular respiration, it begins with the glycolysis, pyruvate dehydrogenase complex, Krebs cycle and the oxidative phosphorylation. In the last session we have discussed the oxidative phosphorylation, in which we have talked about the electron transport chain and the chemiosmotic theory or chemiosmosis. Okay, in electron transport chain we have talked about that it consists of four complexes, that is complex 1, complex 2, 3 and complex 4, and in chemiosmosis there is complex 5 is present. Okay, and this complex 5 is a enzymatic complex that helps in ATP synthesis. Okay, so this complex 5, it helps in ATP synthesis and through the proton motive force. Okay, through a proton motive force. Now the question is, what is the proton motive force? Okay, proton motive force. If you do remember the electron transport chain part, that you remember electron transport chain involves four complexes, complex 1, 2, 3 and 4, where NADH and FADH reduces and it transfers the electron to produce ATP. Okay, so here what happens in this electron transport chain, the complex 1, it pumps four protons from the mitochondrial matrix to the intermembrane space. In complex 3, it exerts four protons, and in complex 4 it exerts 2 protons. Okay, so in electron transport chain there are all total 10 protons are pumped from the matrix to the intermembrane space through the inner membrane of the mitochondria. Okay, and this for one NADH and one FADH. Okay, 10 protons for one NADH, 6 protons for FADH, that we have already discussed. So anyway, when in electron transport chain, the protons are already transported from the matrix to intermembrane space. Now what happens, in mitochondrial matrix, it transported all the positively charged to the intermembrane space. Okay, it transported the positively charged protons to the intermembrane space. So there is lot of protons are present in the intermembrane space. Okay, lot of protons are present. That means the intermembrane is now positively charged. Okay, intermembrane space becomes positively charged, and the matrix, after losing the protons, it becomes negatively charged. Okay, and this is known as voltage gradient. Okay, the positively charged in intermembrane space and the negatively charged matrix, known as this system is known as voltage gradient. When there is more protons, the pH of intermembrane space becomes 7, while the pH of matrix is 8. Okay, and this pH, pH, this is known as the pH gradient. So all together, the pH gradient and the voltage gradient plus voltage gradient is known as electrochemical gradient. So the pH gradient and voltage gradient is known as electrochemical gradient. So the electrochemical potential energy that is created by the proton gradient across the membrane, this is the inner membrane. So the electrochemical potential energy that is created through the proton gradients across the membrane is known as proton motive force. Okay, is known as proton motive force. So complex 5 use this proton motive force to catalyze the ATP synthesis process. Okay, now understood what is proton motive force. So then return back to the complex 5 structure. So this complex 5 helps in ATP synthesis process. That's why it is also known as ATP synthase. Okay, as it helps or it catalyzes the ATP synthesis process, it is known as ATP synthase. So this is also complex 5. Now, what is the location of complex 5 for ATP synthase? In prokaryotic cells or prokaryotic organisms, this ATP synthase lies across the plasma membrane. It lies across the plasma membrane of the cell. In eukaryotic organisms, it lies across the inner membrane of the mitochondria. It lies across the inner membrane of mitochondria. Okay. Also, in the photosynthetic organism, this ATP synthase also present across the thylakoid membrane. It also present across the thylakoid membrane. As example, plant is a photosynthetic organism. Okay, plant is a photosynthetic organism. So plant also contains this ATP synthase in the inner membrane of mitochondria and also in the chloroplast. Okay, because thylakoid membrane is present in the chloroplast, no? So that's in plant has this ATP synthase in inner membrane of mitochondria as well as in the chloroplast. And the cyanobacter, that is also a photosynthetic organism, that also contains this ATP synthase in the cytoplasm. Okay, ATP synthase are present across the plasma membrane in prokaryotes. In eukaryote, it is present in inner membrane of mitochondria. Specifically in photosynthetic organism, it is present across the thylakoid membrane. As example, in plant, it is present in chloroplast, and in cyanobacter, it is in cytoplasm. Okay. Now, if we look at the mitochondria, let's discuss the structure of ATP synthase. Okay, the structure of ATP synthase. So in general, it looks like this in mitochondria. This is the outer membrane. This is the inner membrane. And on inner membrane, there are small tiny structures as present. Okay. This is present all over this inner membrane. So these are the ATP synthase. Okay. These are the ATP synthase or complex 5. Okay. And this is also known as F0 F1 complex. Also known as F0 F1 complex. Why? Because this ATP synthase has two components. One is F0, another one is F1. It has two regions, F0 region and F1 region. So let's discuss both the regions briefly. So somehow this F1 and F0 and F1, it looks like the structure of ATP synthase. It looks like two motors are connected with a central stack. Okay, two motors are connected with a central stack. So if you look at it, one of this region is a rotator. It can rotate, and another where the F1 part can rotate. F0 part can rotate. F1 is a stationary part. Okay. So here this part is the F1 part. Okay. So here this is the F1 part and this is the F0 part. F naught reason of the ATP synthase. Let's discuss one by one the structure of ATP synthase or the F naught F1 complex. So it is known as F naught F1 complex. So first let's discuss the F1 region. Okay, the F1 region of ATP synthase. Now this is the diagram. This is the diagram of F naught F1 complex. This is the F1 part. Okay. So F1 part is present in the mitochondrial matrix. Okay, it lies across the mitochondrial matrix. So reason is it is present in the mitochondrial matrix and this is hydrophilic in nature. This is hydrophilic in nature. Okay. So this F1 contains 5 polypeptide chain. It consists of 5 polypeptide chain. Those are alpha, beta, gamma, delta and epsilon. Okay, it contains 5 polypeptide chain. So here there are 3 alpha and 3 beta. There are 3 alpha and 3 beta forms a ring-like structure. 3 alpha and 3 beta, it forms a ring-like structure that is known as hexameric ring. Hexameric ring, which is the main site of ATP synthesis. This is the main site of ATP synthesis. The mechanism we will discuss later. So now this 3 alpha and 3 beta, it forms a hexameric ring. Then there is gamma and epsilon, which forms a central stack. Okay, the gamma and epsilon forms a central stack which can rotate. Okay, this stack can rotate itself. Okay, and it also connects the F1 to F0 and it runs through the cavity of 3 alpha 3 beta complex or hexameric ring. This gamma epsilon stack runs to the cavity of 3 alpha 3 beta hexameric ring. Okay, and another part subunit is the delta subunit. Okay, delta subunit, it holds the hexameric ring. It holds hexameric ring and rotate it. As I said, and rotate it. Okay. So as I said before that F1 is a stationary part, a stationary region. It can't rotate itself. Okay. So here this delta hold this 3 alpha 3 beta hexameric ring and rotate it. Okay. And the stalk is movable or it rotates itself. Okay. Now this is the F1 region, the structure of F1 region or F1 part. Let's come to the F0 region. As you can see, this is the inner membrane, this is matrix and this is intermembrane space. So F1 part lies at the inner membrane. Okay, it lies across the inner membrane of mitochondria. Inner membrane of mitochondria. Okay, and it contains 9 to 12 C subunits. Okay, it contains 9 to 12 C subunits forming or arranged in a ring structure. Okay, again that is connected to the gamma epsilon stalk and this contains 9 to 12 C subunits which forms a ring-like structure. So these are known as the proton channel. These are known as the proton channel or proton pump, you can say. Okay, it has 9 to 12 C subunits. So these subunits serve as the proton channel. Okay, so when the till the complex 4, the intermembrane space has the proton gradient. So a lot of proton is there. So to neutralize the conditions, some protons flow back to the matrix from the intermembrane space through this channel. Okay, at that time this C subunit acts as a proton channel or proton pump, which allows to transport it to the allows the transportation of proton from matrix to intermembrane space or intermembrane space to matrix to intermembrane space. Here it transfers from intermembrane space to matrix protons. Now there is another subunit, that is A subunit. Okay, A subunit and B subunit. A subunit is connected with the C ring, and B subunit, they form a arm-like structure which connects to the F1 and F0 region. Okay, so if you look at it, it contains 9 to 12 C subunits, one A subunit, one B subunit, the F0 region. So there are 2 points where F1 and F0 they connect with each other. The first one is the gamma and epsilon central stalk, and the second one is the arm that is created with A subunit, B subunit and delta subunit. Okay, so this is the structure of ATP synthase or F0 F1 complex or complex B, complex 5. So F1 region is present in the mitochondrial matrix. It is hydrophilic in nature. It has 5 polypeptide chain, alpha, beta, gamma, delta and epsilon. So the alpha beta is arranged in a hexameric ring, that is 3 alpha 3 beta hexameric ring, and the delta holds it hexameric ring. The gamma and epsilon forms the central stalk. The delta rotate the hexameric ring. Then in F0 region there are 9 to 12 C subunits are present, which forms a ring-like structure, and it acts as a proton channel or the proton pump, which allows the transportation of protons, and there is A subunit and B subunit, which forms an arm-like structure that is connected to the delta subunit of F1. Okay, so this is the structure of ATP synthase. Now we'll see the mechanism of ATP synthase, that how this ATP synthesis takes place. Okay, how this total structure, this machine, you can say it is a molecular machine or enzymatic machine, how this machine going to work to synthesize ATP. So now we'll discuss the mechanism of ATP synthase. So as I said while discussing the structure of F1 portion or F1 region, that F1 3 alpha 3 beta region is the main site. Okay, now discuss the mechanism. So this F1 region has this 3 alpha 3 beta hexameric ring. It has F1 region has 3 alpha 3 beta hexameric ring. That is the main center for ATP synthesis. So it is the catalytic center. Okay. So what happens? This alpha and beta are arranged like a ring. Okay, it arranged like a hexameric ring. So this is responsible for the synthesis of ATP in 3 ways. First, how it is responsible to bind the substrate of ATP. Those are ADP and inorganic phosphate. Okay, first it binds the ADP and inorganic phosphate. It is responsible to catalyze ATP synthesis. It is responsible to catalyze ATP synthesis, and also it is responsible to release ATP. Okay, it is responsible to release ATP. ATP should be released to use for further metabolism process. So it also releases ATP. For the first 2 function, it does not require proton channel. It does not require proton channel, but to release ATP it requires the proton channel, that is the C subunit. Now what happens? If I got the alpha beta, this ring in a horizontal structure, we can see the gamma is running through, so it is the center of it, and there are 3 alpha, 3 beta, no? So let's say it is beta, beta and beta, these are alpha. So alpha has its own bound ATP, it has its own bound ATP. But the problem is it does not release the ATP, neither it takes part in any type of reaction. So alpha does not participate in any reaction, and though it has a ATP, bound ATP, it does not release the ATP. So the main site of ATP synthesis is the beta subunit. Okay, the main site is the beta subunit. So beta subunit binds the ATP, catalyzes the ATP synthesis process and releases the ATP. For all these functions, it has 3 conformational alternates. Okay, it has 3 conformational structural changes. They are known as open state, loose state and tail state. Okay, it has 3 conformational structure to do all these functions. One is open state, loose state and tail state. Okay, so what happens in all these states? See, what happens now when this this F naught part is the movable one, so it always rotates. Okay, so when this F naught part rotates, the C ring rotates. At that time, it also rotates the gamma and epsilon stalk. So it rotates the gamma epsilon stalk. At that point, when the both are rotating, at that point the ATP can be released. Okay, that is only when the ATP can be released from it. So in open state, what happens? Open state always releases ATP. Okay, in open state it releases ATP, and at the same time it binds, so it accepts a set of ADP and inorganic phosphate. Okay, in open state it releases ATP and also at the same time it accepts one new set of ADP and inorganic phosphate to produce ATP. In loose state, this ADP and inorganic phosphate are trapped. Okay, ADP and inorganic phosphate are trapped. That means they cannot leave the site. Okay, in loose state they cannot leave the site, and in tense state or target state, at this state ADP and inorganic phosphate binds together to produce ATP. Okay, so when in tense state this ADP and inorganic phosphate binds together to produce ATP, once ATP is synthesized, ATP is produced, then what happens? This C rings, the gamma stalk, it will rotate 120 degree. Okay, it will rotate 120 degree in anti-clockwise direction. Once the ATP is created, it is synthesized, the gamma central stalk, it will rotate in 120 degree in the counterclockwise direction or anti-clockwise direction. Okay, and when from tense state it is rotating, at that time this beta conformational changes to open state. Okay, when it rotates 120 degree, the tense state converts to the open state. Okay, and in open state it releases that ATP. So open state means it basically open for the transportation of ATP, ADP and inorganic phosphate. Okay, it releases ATP, it also accepts the ADP and inorganic phosphate. This is the only step when ATP is released and inorganic phosphate and ADP is coming. Okay, so once ATP is prepared, the gamma center will rotate in anti-clockwise direction, and here it will produce sorry, here it will release, it will convert to the open state and the ATP will be released. The beta subunit will be relaxed in the open state and it allows the ATP to be released out of it. So this is the mechanism of ATP synthase. So if we look at the order of the reactions, so we can say O state comes first because ADP and inorganic phosphate is coming inside. Then it moves to Ale state, loose state, where ATP and inorganic phosphate are trapped. It can't leave the site. Then the tense or target state, where they bind together to produce ATP, and again it goes back to the O state. Again it goes back to the O state to release the ATP. Okay, so this is the mechanism of ATP synthase. Okay, now this mechanism, this theory is known as binding chains theory, binding chains mechanism. Okay, this theory is known as binding chains mechanism. Just a minute. This theory or descriptive is known as binding chain mechanism. Or flip-flop mechanism, binding change mechanism, or flip-flop mechanism, or flip-flop mechanism. And this theory was developed by Powell Bowen in 1974. Okay, this theory was first developed by Powell Bowen in 1974, which is then supported by John Watson, and then supported by John Watson and James Coe in 1994. Okay, so the first the theory was proposed or described by the Powell Bowen in 1974, but then John Watson and James Coe performed an X crystallography in which they confirmed or they supported the theory. That's why they said Nobel Prize, Powell Bowen, John Watson and James Coe, they said Nobel Prize in 1997. Okay, they said Nobel Prize in 1997. So this is all about ATP synthase. So today we have talked about the ATP synthase, the complex 5, which helps in ATP synthesis in chemiosmosis, and the structure of ATP synthase, that is F naught and F1, that's it is known as F naught F1 complex. It is known as F naught F1 complex or complex 5 or ATP synthase. It has two regions, F naught region and F1 region. While F1 region have alpha, beta, gamma, delta and epsilon. Alpha, beta, 3 alpha, 3 beta forms hexameric ring, which is the main site of ATP synthesis. Gamma and epsilon forms the central star, and the delta holds the hexameric ring and rotate it. Okay, and then the F naught part has 9 to 12 C subunits, which forms a ring-like structure which acts as a proton channel or proton pump, and has A and B, which forms a arc-like structure to connect to the delta of F1, and it connects the F1 and F naught part. And in mechanism, it has the beta subunit has the main role. It is the site of ATP synthesis. So this beta subunit has 3 conformational change, that is open state, loose state and target state or tense state. In open state, it accepts the ADP and inorganic phosphate and releases ATP. Okay, it does both the work. In a loose state, it traps the ADP and inorganic phosphate. In tense state, it converts the ADP and inorganic phosphate to the ATP. And again, that tense state will convert to a open state when this gamma stop will rotate in 120 degree in counterclockwise direction by converting to the beta conformation to the open state, and there this releases the ATP. Okay, that is the mechanism of ATP synthase. So ATP synthase theory is known as binding chains theory or binding chains mechanism or flip-flop mechanism, that is first described or developed by Powell Bower. Then it was again substantiated by John Boxen and Jensku in 1994. That's why they three shared the Nobel Prize in 1997. So this is all about today's discussion. If you like my video, please comment down in the comment box, like and share my video, and don't forget to subscribe my channel. Thank you so much.